And we have gotten to the section where we were going to talk about mutations. So I've mentioned before when we talked about haploid and diploid and mutations, and a mutation is going to be a change in that DNA sequence. So when a mutation happens, the DNA changes. When that happens, the RNA is going to change, depending on if it's in an mRNA, if it's in a gene, the RNA can change. And then we can see how the codon, if that RNA codon changes, how it's going to give rise to a different, could give rise to a different amino acid, and that's going to put a different protein, a different amino acid in a protein, and that can change the shape of a protein. And we've seen how specific this binding is between protein and protein, and so that can make a big difference. So one change in the DNA can make a huge difference, potentially can make a difference in a person or in a host or in a person, and that can cause a disease. So there are lots of diseases caused by mutations. So all cells that have DNA have mutations, and so bacteria said sometimes they get mutations and they die, and no big deal because there's others. So let's talk about mutations. Now, normally, if you change the shape of a protein, something bad's going to happen. It's not going to work. If I were just to change something, it's probably not going to work. But there are cases where sometimes some of these mutations actually improve the function of a protein, and if they improve it, then that's going to give that particular cell a survival advantage. So sometimes mutations can be beneficial. Most of the time, no. But sometimes you get a change and it makes it better. And that can lead to a survival advantage. And that's when we're talking about evolution, that things evolving. It's got a slightly different shape. It gives it a better outcome, survival advantage, which means it can live and reproduce, and then those progeny also have that survival advantage, and it moves forward with that. All right, so let's talk about mutations at the DNA level. All right, so it says change in the base of a gene. It's a rare event. Almost always it's a bad thing. But sometimes it can improve the organism's ability to survive. Now, we can put mutations in different categories. Sometimes there are big mutations, like an entire chromosome gets deleted or replicated, or parts of a chromosome. Those are considered gross mutations. They're almost always lethal, but more likely is one nucleotide is affected, and those are called point mutations. They're the most common types of mutations where you just get one change in one nucleotide. It affects one base pair, and some of these do fine. So let's talk about different categories of point mutations. And again, only one nucleotide is affected. Different ways that nucleotide can be affected. You might get a substitution. So it's supposed to be a G, and it gets substituted as a T. That is one type of point mutation, a substitution. Another type of point mutation, again, only affecting one nucleotide, is an insertion or a deletion. You get an insertion, it's supposed to be AUG, and it becomes AUUG. You get an extra nucleotide inserted, or a deletion, AUG becomes AG. You get a deletion of a nucleotide. All of these are still considered point mutations because they only affect one nucleotide. Now, you can categorize these mutations based on their outcomes. When you have an insertion and deletion, you're going to end up with a frame shift. We'll talk about that in a minute. If you have a substitution, you're going to end up with a different set of outcomes, and those are going to end up being silent mutations, missense mutations, or nonsense mutations. Let's skip to the next slide, and let's look at these in more detail. It'll help you understand it. Okay, so this should look familiar. This is your codon chart. You know how to read it. This up here is your normal DNA sequence. So this is what's normal. This is what you've been passed down. You've got your DNA that transcribes into an RNA sequence, and then it gets translated into a polypeptide chain, which makes a protein. And in this case, you get phenylalanine, tyrosine, alanine, and arginine. So you get a sequence polypeptide, and then that protein is going to form and make a shape. Let's say there's a mutation in this. So instead of the DNA sequence AAA, you end up with a substitution. And now instead of AAA, it becomes AAG. So we're going to look at substitutions and how they may affect the phenotype. The genotype is changed, I have a different nucleotide here, so I have a different genotype. Let's see if that affects the phenotype. All right, so AAA triplet gives rise to the UUU codon. In this case, with the mutation, we have a UUC. So UUU, here we go, UUU gives us phenylalanine. Okay, so that's good. UUU gives us phenylalanine. What happens with the mutation? Instead of UUU, we have UUC. UUC, what do we get? We still get phenylalanine. So the protein, the amino acid sequence remains the same. There's no change in the amino acid sequence, which means the protein looks just like normal, and there's no change in the protein. The phenotype is the same, even though the genotype changed. This is just a variant, not a big deal, and we refer to this as silent mutation. If it makes the same protein, even though the way you spell it is different, it's called a silent mutation. And these don't have any effect at all because it's the protein that does the work. To compare it to something that you can, think about a little better, if I have a sentence that says cow eats grass, okay, that's great. That makes sense. Cows do eat grass. I can read that sentence, no problem. If I get a mutation and I switch the C in cow to a K in cow, cow eats grass. This one you had two mutations. I actually can still read this sentence, right? I speak Chick-fil-A. I have no problems reading this sentence. Cow eats grass. It still makes sense. Cow eats grass. Sometimes you just get a different spelling, but you get the same outcome. This is an example of a silent mutation. So it's a substitution, and the particular category of substitution is a silent mutation. Let's look at another one, though. So here, instead of, let's see, AAA, ATT, here, so we get a mutation here, AAA, ATA, that leads to a codon, and instead of, let's see, you get UAA, instead of getting tyrosine in the second spot, you get UAA, UAA, instead of giving you tyrosine, you get a stop codon. Remember what happens in a stop codon? The ribosome stops translating. So instead of this polypeptide chain with phenylalanine and tyrosine and alanine, ooh, I skipped one, didn't I? I went right here. That's okay. We'll do this one next. I went right to phenylalanine and then a stop codon. So in this case, do I have the same protein that I'm supposed to make? No, I have put a stop codon. This is called a nonsense mutation. If I do it back to here, instead of cow eats grass, I just have cow. Cow and then stop. Well, that makes no sense. What about cows? Are they black? They eat grass? I mean, I don't know. It's just cow. It stops. This is a nonsense mutation, and this potentially has a problem for creating a disease. This protein is not made, and it can't do its job if it doesn't exist or if a piece of it exists. So nonsense mutations can lead to disease. Now you might get lucky that stop codon may be real close to the end and you can still make it by, but these have a higher chance of having problems. All right, so I skipped missense mutation. Let's go back to that one. So I have one substitution. Instead of a C here, I get a G. If I go to the codon chart, instead of getting an alanine in this third position, I end up with a glycine. So I get a change in the amino acid sequence. So here, instead of putting the alanine, I got glycine, I get a different word here. Phenylalanine, tyrosine, glycine instead of phenylalanine, tyrosine, alanine. So this would be similar to this. Instead of cow eats grass, I get a change, and now it says car eats grass. Well, it's still a sentence, but the sentence doesn't make sense. It's not doing the job it's supposed to do. Cars don't eat grass. That doesn't even make sense. So a missense mutation is when you get a different amino acid substituted, and then that's going to change that protein. These can also lead to diseases, depending on where it is. Maybe you get lucky and it's in a place that it doesn't cause a problem. Maybe it's in the active site, and this is right in the middle of an active site, and it's going to stop that protein from doing its job. So missense mutations can lead to disease. Okay, all right, so these are the three types of mutations we get when we have a substitution, and we'll go back to our summary slide. Substitutions can cause silent mutations. The base pair is substituted, but you end up with the same amino acid. It can cause missense mutations. The base pair is substituted, and you get a different amino acid in that protein, or a nonsense mutation, and that's where you get a substitution and it encodes for a stop codon, and you get a truncated protein or a shorter protein. So these are the types of mutations that you have if you have a substitution. Let's look at what happens if you have either an insertion or a deletion. So let's start with my sentence here. I've got a series of three words that make up a sentence. 3-letter words that make up a sentence. I have the fat cat ate the big rat. That makes sense. No problem. You can read it. What happens if I delete the T? Now remember, if this is a nucleic acid, we don't have placeholders or anything. If we delete the T, then instead of FATC, if I delete the T, it's FAC. This is going to make a big difference because the ribosome is just reading from one side to the other. So it starts reading THE, that makes sense, and then the T is gone, fat, so we have FA, and it skips to the next one, C, ATA, ATA, TET, and you can see this caused a shift. All of the letters moved up, and instead of this nice sentence that makes sense, we have something that's gibberish that makes no sense. This happens if you have a frame shift mutation. Either an insertion or a deletion will cause a frame shift, and it means that every amino acid read after this is going to be off by a nucleotide, and then you're going to end up with a completely different protein. These also give rise to issues or a change in phenotype, because not only did you not make the correct protein, but you made this weird protein that who knows what it's going to do. So not only are you missing the correct protein, but you've made some strange protein that could interfere. So insertions and deletions give rise to frame shift